Hello everybody, we are gonna talk about the difference between innate and adaptive immune system. My name is Putri Indra Niagari. Here I am with my friend Rachmadya and Icthiarie. We are from RA class of program study BIOTEKNOLOGY in Brawijaya University. Okay, here I'm gonna explain about the immunity. Immunity is defined as resistance to disease, specifically infectious disease. Our immune system is consist of two basic types of immunity. The first is innate or natural immunity and the second is adaptive or acquired immunity. I'm gonna start from innate immunity. And then now, let's focus on innate immunity. Innate immunity is also called as natural immunity or native immunity. Why is it called as natural immunity or native immunity? Because it's always present in healthy individuals to block the entry of microbes, to rapidly eliminate microbes that do succeed in entering host tissues. Innate immunity is the critical first step in host defense against infections. Innate immunity is the first line of defense provided by epithelial barriers of the skin and mucosal tissues and by cells and natural antibiotics present in epithelia, all of which functions to block the entry of microbes. It blocks microbial infection through epithelial barriers, destroy many microbes that do enter the body, and is capable of controlling and even eradicating infections. There are 7 general features and specificity of innate immune response. The first is the two principal types of reaction of the innate immune system are inflammation and antiviral defense. And the second is the innate immune system responds in essentially the same way to repeat encounters with a microbe. And the third is the innate immune system recognizes structure that are shared by various classes of microbes and are not present on normal host cells. And the fourth is the components of innate immunity have evolved to recognize structure of microbes that are often essential for the survival and infectivity of these microbes. The fifth is the innate immune system also recognizes molecules that are released from damaged or necrotic host cells. And then the sixth is the receptors of the innate immune system are encoded by inherited genes that are identical in all cells. And then the seventh is the innate immune system doesn't react against the normal host. The innate immune response can be considered as a series of reactions that provide defense at the following stage of microbial infections. The first is at the portals of entry for microbes, and the second is in the tissues, and the third is in the blood, and the fourth is viruses elicit special reaction. These are the components of innate immunity. They are epithelial cells, sentinel cells in tissues, innate lymphoid cells, and plasma proteins. There are 3 functions of epithelial barriers in immunity. The first is sickle barriers to infections. Epithelia presence at the portals of entry of microbes provide sickle barriers formed by keratin in the skin or secreted mucus. And the second is killing of microbes by locally produced antibiotics. Epithelial barriers also produce antibiotics to kill microbes. And the third is killing of microbes and infected cells by intraepithelial lymphocytes. Intraepithelial lymphocytes, which function is to prevent microbes enter our body by killing microbes and infected cells. Neutrophils and monocytes are blood cells. They are required to sites of infections where they recognize and ingest microbes for intracellular killing. Neutrophils. Neutrophils are the first cells type to respond to most infections, particularly bacterial and fungal infections, and those are the domain cells of acute inflammation and as discussed later. And the second is monocytes. Monocytes or macrophages serve several important roles in host defense. They produce cytokinase that induce and regulate inflammation. They ingest and destroy microbes, and they clear dead tissues and initiate the process of tissues repair. Dendritic cells respond to microbes by producing numerous cytokines that serve two main functions. They initiate inflammation and they stimulate adaptive immune response. And the fourth is mast cells. Mast cells can synthesize and secrete lipid mediators and cytokines, which stimulate inflammations. Innate lymphoid cells. Innate lymphoid cells are lymphoid-like cells that produce cytokines and perform functions similar to those T lymphocytes but don't express T-cell antigen receptors. And natural killer cells. Natural killer cells recognize infected and stressed cells and respond by killing these cells and by secreting the macrophage-activating cytokine IF and gamma. NK cells kill host cells infected by intracellular microbes, thus eliminating reservoirs of infections. And then the B is NK cells respond to interleukin 12 produced by macrophages and secrete interferon gamma, which activate the macrophage to kill phagocytic microbes. Plasma proteins such as CRP is a pentraxin that binds to phosphorylcholine on microbes and opsonizes the microbes for phagocytosis by microbes, which express a receptor for CRP. CRP can also activate protein of the classical complement pathway. The circulating levels of many of plasma protein increase rapidly after infections. There are two kinds of innate immune reactions. They are inflammatory response and antiviral defense. In inflammatory response, the macrophages, dendritic cells, mast cells, and other cells produce cytokines and other mediators as a response to microbial products and damage host cells. The permeability of blood vessels is increased by the mediator, leading to the entry of plasma protein into the tissues and promote the movement of leukocytes from the blood into the tissues, where the leukocytes destroy microbes, clear damaged cells, and promote more inflammation and repair. Type 1 interferons are produced by plasmacytoid dendritic cells and virus-infected cells in response to intracellular TLR signaling and other sensor of viral nucleic acid. Type 1 interferons bind to receptors on the infected and uninfected cells and activate signaling pathway. They induce expression of enzymes that interfere with viral replication at different steps, including inhibition of viral protein translation, increasing signal RNA degradation, and inhibition of viral gene expression and variant assembly. Type 1 IFN hinge the ability of NK cells to kill infected cells.
So now I would like to explain about adaptive immunity. What is adaptive immunity? Adaptive immunity, or also known as specific or acquired immunity, is composed of highly specialized systemic cells and processes that eliminate or prevent pathogenic growth. The adaptive immunity works to protect and heal the body when the innate immune system fails. The adaptive immune system consists of lymphocyte and its products, for example, antibody. Every ingredient it can be specifically recognized by lymphocyte, and antibodies are called antigens. Response adaptive immunity using cells as well as molecules from the natural immune systems to eliminate microbes, and function adaptive immunity to assess to antimicrobial immunity already. For example, antibodies binds to microbes, and microbes that serve antibodies it bind strongly to existing phagocytes activated, and activate these phagocytes that is digest and destroy microbes. There are two types of adaptive immunity. There are humoral immunity and cell-mediated immunity. First, I will explain about humoral immunity. So, humoral immunity. What is humoral immunity? Humoral immunity is antibody-mediated immunity produced by B lymphocytes. And what is B lymphocytes? B lymphocyte is any of the lymphocytes that have antigen-binding antibody molecules on the surface that comprise the antibody-secreting plasma cells when mature and that in mammals differentiate in the bone marrow B cell. And what is antibody? Antibody is a protective protein produced by the immune system in response to the presence of a foreign substance called an antigen. So this is 3 facts about B lymphocytes. B lymphocytes are formed and mature in the bone marrow. B lymphocytes have receptors which antigens can attach. If there are antigens attached to the receptors, it will stimulate B lymphocyte to turn into plasma cells. Plasma cells will produce antibodies. And now about antibodies. Antibodies are glycoprotein compounds that have a curtain structure in the form of a response to curtain antigens. Antibodies are secreted by activated B cells into plasma cells. When a foreign object enters, it takes between 10 and 14 days for the substance's antibody to form completely. And now we will discuss about cell-mediated immunity. Cell-mediated immunity is the host's primary response against invasive bacteria and viruses that cause intracellular infections. It is also essential for destroying cancer cells and rejection of organ transplants or graft tissues. Cell-mediated immunity produced by direct action of immune cells such as T lymphocyte. And now we will discuss about T cells. There are 4 types of T cells, namely cytotoxic T cells, helper T cell, regulatory T cell, and natural killer T cell. Cytotoxic T cell is involved in the direct destruction of cells that have become cancerous or infected by viruses. Helper T cell accelerates the production of antibodies by B cells, produce substances that activate cytotoxic T cells and macrophages. Regulatory T cells suppress the response of B cells and T cells to other antigens, and natural killer T cells distinguish infected or cancerous cells from normal body cells and invading cells that don't contain molecular markers that identify them as body cells. And this picture is about the difference of B lymphocyte, helper T lymphocyte, cytotoxic T lymphocyte, and regulatory T lymphocytes. There are 3 types of immune system cells, namely lymphocyte, antigen-presenting cells, and effector cells. Lymphocyte include B lymphocyte and T lymphocyte. B lymphocyte is mediator of humoral immunity, and T lymphocyte is the mediator of cellular immunity. And then antigen-presenting cells, including dendritic cells, macrophage, B cell, and follicular dendritic cells. Dendritic cells is the cells that initiate the T cell response, and macrophage is the effector phase of cellular immunity, and then follicular dendritic cells is show the antigen to B lymphocyte on the humoral immunity, and then effector cell including T lymphocyte, macrophage, and granulocyte. T lymphocyte is the cells to activate the phagocyte, and then macrophage is to kill the microbes, and granulocyte is same with macrophage. The function is kill the microbes. This picture is the difference between humoral immunity and cellular immunity. In the humoral immunity, the types of microbe that recognized by lymphocyte is extracellular microbes, while in the cellular immunity is the intracellular microbes. And then the type of lymphocyte that give a response is B lymphocyte in the humoral immunity and T lymphocyte in the cellular immunity. And then the mechanism of humoral immunity is where the antibody defends infection and kill the extracellular microbes. And the mechanism of cellular immunity is where the helper T lymphocyte eliminate microbes in the phagocytosis, and the cytotoxic T lymphocyte will kill the infected cell and kill the infection of reservoir. This picture is the difference between innate immunity and adaptive immunity. Adaptive immunity takes several days until the antigen can be destroyed by antibody or effector T cells.